Everybody. First, I want to thank Jennifer, Mathieu, and Joseph for putting together this conference and for inviting me to speak. I'm very excited to be here today and during this week. I think it's a great opportunity to discuss synaptic plasticity with people with various backgrounds, so thank you for this. So I'm a cellular neurobiologist, so I'm really focused on how cells behave and how they communicate with their environment. And today, I would like to discuss some recent work from our lab in Bordeaux, and in which we show that individual connections, individual synapses, like synapses in between two neurons, so I'm talking about unitary connections, compensate for chronic inactivity by increasing their strength to achieve homeostasis. And I'm going to try to convince you that this synapse autonomous regulation is going through the local translation of a protein that is called Synaptopodin. And I'm going I'd like to discuss with you the possibility that Synaptopodin serves as a synaptic tag to provide autonomy to synapses and also to provide plastic properties to synapses. Okay, let me first introduce briefly what we are doing more generally in the lab, what are the type of questions we are trying to address. So we are trying to understand how neuronal activity shape neural connections in central circuits. So this is obviously a difficult question if you consider the complexity and the diversity of synaptic circuits, which I think is well illustrated here with this 3D reconstruction of a saturated volume of cortical circuits. So if you take individual neurons in such a crowded environment, they are probably surrounded by hundreds of different cells, but they make contacts and connections with only a fraction of them. So what's the molecular mechanism driving this specific connectivity, and what's the contribution of activity? So the question is even more complex if you consider the diversity of synaptic connections that can be made between two neurons. If we take the examples of dendritic spines, you can see that they exhibit various shapes falling into different categories: mushroom spines, filopodial protrusions, stubby spines, thin spines, exhibiting different functional properties. So one way to quantify the diversity of those connections is to plot the distribution of synaptic parameters, including spine size, frontal size, and release probability at individual synaptic contacts. And if you do that, you will see that all those parameters follow a log normal distribution, showing a large range of values and a skewed shape. And what we want to understand is how neuronal activity or synaptic activity coming from different synaptic inputs shape this distribution, and what's the mechanism for that. So of course we know that synapses are plastic, right? So that's why we are here today, so they can grow in size, they can get stabilized through LTP-like mechanism, or in the other way around, they can shrink, get weaker, and eventually get eliminated through LTD-like mechanism. We also know that neuronal activity may play a role during synapse formation, during development, or learning and memory processes. And our way to try to approach the role of synaptic plasticity in the diversification of synaptic connection is to characterize individual connections between individual cells. So we are developing single-cell labeling and manipulation strategies using patch clamp pipettes, electrodes. And this is really the effort we are trying to make in the lab, is to correlate synaptic parameters through electrophysiological recordings, and at the same time to get insight into the structural dynamics of this the interaction made between these two cells, to get insights in the molecular organization using high resolution microscopy, and in this case expansion microscopy, and also to get some correlative results on gene expression, so how activity can affect gene expression in each of the two neurons. So we are implementing with one of my colleagues in Bordeaux the patch-sec technique. So this is the general frame of our research. That's what we are trying to do to understand better, to get insight into the diversity of synaptic connections by using correlative approach. So which we think in terms of statistics is highly relevant. So actually, I started to use this single-cell labeling approach while doing my postdoc in the lab of Yuki Goda several years ago. And what we showed by labeling individual connections is that synapses, that synaptic contacts that share the same presynaptic neuron are more similar in comparison to synapses that are established by different presynaptic inputs. So we could see that by measuring spine size, AMPAR receptor synaptic content, or release probability at individual synaptic contacts. So in each case, the disparity is decreased if you compare synaptic contacts made by the same presynaptic neurons. So clearly, there is the identity of a presynaptic cell will set the structure and the function of individual connections. So we also showed that you can promote the disparity between distinct inputs by eliciting classical LTP using a high-frequency stimulation protocol, and we also showed that at non-stimulated connections you can get heterosynaptic plasticity, and this heterosynaptic interaction that promotes the disparity between the inputs involves the associative calcium signaling involving NMDA receptors and LTAP calcium channels, and we were able to show by infusing pharmacological brokers in the associative syncytium that the disparity get decreased if you block this calcium signaling. And similarly, on the postsynaptic side, it was shown that spines also undergo similar heterosynaptic interactions through calcium signaling. So this is a work from Karen Zito's lab showing that if you induce LTP through glutamate and caging at a bunch of spines, here you can get heterosynaptic shrinking. So clearly activity promotes the disparity between distinct inputs. Now I would like to introduce the mechanism that could maintain this disparity in the very long term, because this will be useful for the rest of my presentation. So one model is the synaptic tagging and capture hypothesis proposed by Frey and Morris in 1997. Showing that, so what they propose is that potentiated synapses are molecularly tagged by one or maybe a set of molecules. And if the depolarization is strong enough, then a signal to the nucleus will be sent to promote gene expression, the synthesis of plasticity-related products that will be then trafficked and captured by the molecular tag specifically. So this is a powerful approach to, a powerful model to explain the long-term maintenance of potentiation at specific synapses. An alternative model that is not mutually exclusive is the local translation that has been shown to occur locally, so at proximity of spines or in the axon. And this could be a way also to fuel synapses with new proteins to maintain changes in the long term. So we know that all the translation machinery, including ribosome, messenger RNAs, and translation regulators, such as microRNAs and RNA binding proteins, can be found at proximity of synapses, and that they are regulated by synaptic activity. Okay, so at this stage, I would like to moderate a little bit the role of activity in creating contrast across synapses by mentioning some work in which, so especially from Tom Sudov's lab and Niels Bro's labs and also Anton Maximov's labs, showing that if you impair glutamate release from synapses or neurotransmitter release by using genetic model where presynaptic proteins are knocked out or where you express tetanus toxin in the presynaptic terminal, so to block the release, synapses can still form and exhibit rather normal ultrastructure, and they can get maintained, and they also exhibit a high diversity. So this is an example here showing dendritic spines in the control situation or in a model where there's no glutamate release, and you can see that those spines exhibit all the repertoire of morphologies that I mentioned before. They are functional and they are in similar amounts. So this clearly questions the role of synaptic transmission in for synapse assembly and for the diversification of synapses. So from now, I would like you to keep those results in mind and to think them in terms of homeostatic plasticity that was not really discussed in those papers. So what is homeostatic plasticity? It's a type of plasticity in which neurons compensate for chronic inactivity by increasing the strength of their synapses and by increasing the size of their synapses as a way to maintain or to restore a normal spiking rate. So here the parameter that is sensed is the firing rate. And this was first described by the group of Gina Torrigiano and Enrique Gagné in the late 90s using cultured neurons and pharmacological blockade of activities such like TTX or CNQX and AP5 to block action potentials on glutamate receptors. And later on, it was also shown to occur in various physiological or pathological situations, also in vivo, for instance, following sensory deprivation. So one key feature of this plasticity that was proposed is that every synapse on the neuron is scaled with the same factor, which led to the concept of multiplicative synaptic scaling, which means that you can linearly transform the distribution of synaptic strength from the basal condition to the activity-deprived situation by multiplying each individual values by the same factor. So to sum up, we have a type of plasticity which provides a negative feedback to maintain the firing rate, which involves cell-wide and global signaling affecting every synapse simultaneously, and which doesn't depend on prior synaptic activation since every synapse is scaled with the same, in a similar way. And finally, this type of plasticity should maintain relative synaptic strength differences, which is really important because it means that there is no transformation of information stored across synapses. So this is, of course, in contrast with LTP, which is also leads to the strengthening of synapses, but this time in response to acute synaptic hyperactivity. So we have two very different induction protocol. LTP relies on positive feedback mechanism. It displays input specificity, as I mentioned before, and it's influenced by prior synaptic activation, meaning that not every synapse will exhibit the same ability to undergo the potentiation. And finally, it's expected to alter synaptic strength distribution, as discussed before, to store new information. So we have two opposite types of plasticity, one, but that can coexist because one is occurring at a large scale to maintain the firing rate, and one is occurring at the local scale to store new informations. Yes?
You get downscaling if activity is too high as well, right? Or...
Exactly. So this is a bidirectional, homeostatic plasticity can be bidirectional, but today I'm just focusing on the scaling up process. So from now, I would like to challenge a little bit this idea, and this is what I'm going to do until the rest of my presentation. And I will start by highlighting a few points. The first one is that these two types of plasticities converge to common signaling pathways and molecular actors, which are highlighted here, and they both rely on protein synthesis. The second aspect is that there is a few studies showing that homeostatic plasticity can be expressed in subcellular compartments independently from the cell body, and this is one example here, where individual presynaptic terminals were silenced by overexpressing the KIR2.1 channels. And what the authors found is that the postsynaptic spines exhibit larger currents through the expression of a GluA1 AMPA receptor subunit, so suggesting a local regulation of homeostatic plasticity that could be supported by local translation mechanism that has been described also and shown during homeostatic plasticity in neurons treated with TTX. So this is an example from Erin Schumann's lab using metabolic labeling of newly synthesized proteins in magenta here, and you can see that following TTX treatment and just after 1 hour of treatment, there is a large increase in the translation rate of proteins in the synaptodendritic compartments, and this increase lasts for at least 24 hours. So in one possibility, this local protein synthesis could tune synapses individually in a non-uniform manner. And actually in the lab several years ago, we showed that the GluA1 subunit of AMPA receptors can be locally synthesized in dendrites, and that this translation, this synthesis is regulated by a microRNA, miR92A, which normally binds the 3' UTR of the transcript to inhibit the translation, right? And what we found is that this microRNA is downregulated upon activity deprivation, which leads to GluA1, which derepress GluA1 translation, and promotes the insertion at synapses and the strengthening of synapses. And we found that if you overexpress this microRNA to compensate for the downregulation, there is a blockade of the homeostatic response. So all this led us to this emerging view, and to propose this emerging view that synapses may detect their own activity rather than the spiking rate, at least in some situation, and self-regulate their strength through local resources. And the second point is that not all synapses have the ability to homeostatically adjust their strength. So these were our hypotheses, and we decided to study Synaptopodin as a molecular handle to get further into these ideas. And I'm going to explain why Synaptopodin. So Synaptopodin is an essential component of an organelle that is called the spine apparatus, which is emanating from the endoplasmic reticulum here, and form this stack of cisterna from the endoplasmic reticulum. So importantly, the spine apparatus is expressed in only 10% of spines in telencephalic neurons. And Synaptopodin is an essential component, meaning that if you knock out Synaptopodin, the spine apparatus will not form. The second point is that Synaptopodin and the spine apparatus are associated with strong and stable synapses. Those 10% synapses are the strongest one. They mediate the largest currents across the neurons. And if you knock out Synaptopodin, you have a strong impairment in both LTP, but also homeostatic plasticity induced by presynaptic denervation or TTX treatment. So this puts Synaptopodin as a very critical actor in these two types of plasticity, probably acting through the actin dynamics or calcium homeostasis within the spine, or by regulating receptor trafficking. And the last point is that Synaptopodin might be synthesized locally, considering these few evidence. One is that there's no obvious transport of Synaptopodin when doing live cell imaging. So the Synaptopodin puncta seem to appear directly within spines. Another evidence is that when you perform LTP in acute slices, Synaptopodin immunoreactivity appears directly in the stimulated layers, not in the cell body layer, for instance, suggesting a local synthesis of Synaptopodin. And the last point is that we have shown previously that Synaptopodin translation is regulated by a microRNA, which is miR124. It's a very abundant microRNA in neurons, which is present in dendrites, and which has been shown to regulate both LTP and homeostatic plasticity. So the rationale to look at Synaptopodin was rather strong, and then we decided to address a series of questions. The first one was, do Synaptopodin positive and Synaptopodin negative spines adapt similarly to global activity deprivation? Sure.
So is the spine apparatus made up of Synaptopodin, or is Synaptopodin protein just kind of hangs out on the active skeleton?
So Synaptopodin is part of a dense matrix that is found in between the saccus of the spine apparatus. We don't know exactly how it allows the formation of the spine apparatus. This is not really known. This remains mysterious. And actually, we don't know much about the molecules that are present at the spine apparatus. There is a recent proteomic study from the Camille lab, but there's not much known in terms of molecules and the spine apparatus. What is very clear is that if you knock out Synaptopodin, there's no spine apparatus that can form. So probably it's involved in the shaping of the spine apparatus somehow, maybe involving the actin cytoskeleton, because we know that there is a direct interaction between Synaptopodin and alpha-actinin, for instance.
So
but,
there are binding motifs.
Yeah. Yeah.
Just wondering, is it 10% synapses that are stable, or is it like, are those the same as speed statements?
So the 10% of synapses which contain Synaptopodin, they are the strongest ones. So if you compare Synaptopodin positive and Synaptopodin negative spines, for instance, doing glutamate and caging, or just looking at the turnover the synaptopodin positive ones. So presumably having a spine apparatus will be, you will find them much more stable and stronger than the others. So there is really a correlation. So do synaptopodin positive and synaptopodin negative spines adapt similarly to activity, global activity deprivation? We would expect that if we have a uniform homeostatic scaling, but we wanted to test that. So for this, we performed immunostaining experiments in culture neurons transected with HOMER1C GFP in magenta here to visualize synapses, and we immunostain endogenous synaptopodin and AMPAR receptors. So you can see that synaptopodin immunoreactivity is found at large synapses. I don't know if it's really clear here, but it's only in a fraction of large big synapses. And when you treat neurons with TTX for 48 hours, which is a classical paradigm to induce the plasticity, we have a large increase in the percentage of synaptopodin positive synapses, together with an increase in the average synapse size and an increase in the average AMPAR receptor synaptic content. When taking a closer look, what we found is that the increase in AMPAR receptor synaptic content was larger for synaptopodin positive and large synapses than for synaptopodin negative and small synapses, suggesting that the large and the synaptopodin synapses, positive synapses, contribute more and respond better to the activity deprivation protocol. We could very, yeah. Yeah, sorry, I'm not sure I understood completely the question.
The question is the chicken and the egg, right? So if you don't have synaptopodin, you don't have this effect homeostatically, and you don't have LTP because you said you need to have it. So how? So then 90% of your synapses are doing nothing and they're unrescuable.
Sorry, I'm not sure to really get what you. So you need a synaptopodin to get
have LTP.
Yeah.
And you need it also to have this
exactly.
form of. And so if you don't have it, how you ever the 90% of synapses that don't have it, how do they ever get to play?
That's a key question. So what are the plastic properties of these synaptopodin negative synapses? So this we didn't really investigate that. In terms of this homeostatic plasticity, it seems that they do not respond. For LTP, I'm not sure it was really investigated. At the single synapse level. So there was no discrimination between synaptopodin negative and synaptopodin positive synapses. So I think that's a critical experiment that actually we are planning to do. But in the literature, the way it was investigated for LTP, just using field stimulation. So we cannot discriminate between the two. And at the level of field stimulation, you get an impairment of LTP, suggesting that that synapses deprived of synaptopodin do not, are not plastic. But this would need to be confirmed.
Can you go back one slide? At the risk, no, sorry, go to your synaptopodin next.
Yep.
Next.
Again?
That one. So at the risk of being reviewer 3 here, it's much easier to measure a change in a large synapse than it is to measure a change in a small synapse. So you do have what looks like a change. It is not statistically significant.
Yeah, I agree.
But I have troubles thinking maybe that this is actually the fact that it's easier to measure it in the big synapses. It's not that it's not happening. Maybe it's happening to a greater degree.
Yes, yeah, that's for sure. And I wouldn't say that there is no increase. I wouldn't say so. I would say that the extent is probably less. Then we have, so in this case, we have, this is unpaired data, okay? We are comparing untreated and TTX situation. Later on, I will show you preliminary preliminary data in organotypic slice where we look at the very same spine before and after. And I think this is more convincing because this is one bias that we could have here, I agree. So we'll come back to that. But in any case, we have an increase in the percentage of synaptopodin positive synapses, meaning that they do not all respond similarly. And this is really binary expression of synaptopodin. So the conclusion here was that the plasticity is not uniform. Okay, so this is what those results suggest. And we could also confirm that by plotting the distribution of AMPA receptor synaptic content between the two conditions, we were not able to find a linear relationship between the two distributions. So if you look at the distribution of the untreated and the TTX condition in light blue and dark blue, you can see that there is an overlap for the smaller values, suggesting that, again, the small synapses respond less or contribute less to the plasticity than larger and stronger synapses. And we got similar results looking at miniature excitatory postsynaptic currents. We got exactly the same. So we got the same average increase in the size of the response. And then when you plot the distribution of quantile events, you don't see a multiplicative increase. And you see again that in the small values, you have an overlap of the two distributions. And here it's, I mean, there might be a problem of sensitivity, you're right, even for the small events. But here we have two different approaches, immunocytochemistry and electrophysiological recordings that converge to the same conclusion. Okay, so the next question was, is synaptopodin locally translated upon activity deprivation? Because it seems that we have synapses that become synaptopodin positive. Where does this synaptopodin coming from? So to address that question, we used an assay. Yes.
I think maybe you said it, but like on a molecular level, like what is regulating the synthesis of this synaptopodin?
I will come back to that right after. So this is part of the. So, but first we wanted to know where is synaptopodin synthesized within the neuron, and for this we used pyromycin proximity ligation assay, which has been previously used by the Schumann's lab. And so the idea is to incubate neurons with pyromycin that will, for a short period of time, it was 5 minutes in our case. So this pyromycin will integrate nascent peptides and terminate the translation and get incorporated into the nascent peptides. And then so then you fix the neurons and then you can process them for immunostaining using one antibody against pyromycin and one antibody against an epitope of your protein of interest, so in our case, synaptopodin. And because of the proximity between the two antibodies, you can generate a specific signal by using PLA probes conjugated to oligonucleotides. So you can get a very high amplification of the signal and get a very high signal-to-noise ratio and detect individual newly synthesized molecules. So we did that for synaptopodin, and as you can see here, we got a very stark increase in the number of clusters where translation occurs, and in the intensity of fluorescence intensity of this cluster, suggesting more translation sites for synaptopodin and a higher rate of translation following the TTX treatment, and this is quantified here. And we also found that synapses in which the translation is occurring, they are more numerous following the TTX treatments, and they are also higher in size. So if you look at these individual synapses, they seem to be larger compared to spines or synapses where the translation of synaptopodin is not taking place. So this supports the idea that the translation of synaptopodin enables the growth and the strengthening of individual synapses. So then we wanted to ask whether this process was...
Sorry, question? Yeah. I think I just may have missed it. Is that synaptopodin synthesized in synapses that already have it and it's enhanced, or also in synapses that don't have it at all?
That's a good question. I think that's both, actually. So I will show you images after where we see the same spines before and after, and it seems that some spines start expressing synaptopodin, so they were devoid of synaptopodin, and others express even more synaptopodin. So I think it's both.
Thanks. I may have missed this also, but this proximity ligation assay shows you newly synthesized protein, but this is 48 hours of TTX, right? So it could have been synthesized anywhere and transported to the spines. So how do you actually have evidence that it is synthesized at those locations where you see it now?
Yeah, you're right. So the TTX treatment is 48 hours, but the pyromycin incubation is just 5 minutes. So you incubate neurons with pyromycin, and the pyromycin will get incorporated to the peptides within these 5 minutes. And I mean, this, of course, you can always think that during these 5 minutes there will be a transport of a protein, but I mean, I think it's okay. So then we wanted to look at whether synaptopodin could be regulated by miR-124, and it's coming to your question, I think. And one reason to look at miR-124 is that because this microRNA is really enriched in neurons, and it has been associated with both LTP and homeostatic plasticity previously. And so our hypothesis at that point is that some synapses in the basal condition are deprived of synaptopodin. There's no spine apparatus. But they have at their proximity the messenger RNA for synaptopodin bound to the microRNA 124. This was our hypothesis. So that's why they are, in fact, they are deprived of synaptopodin, but ready to express synaptopodin under a specific stimulation. And so the hypothesis was that following 48-hour CTX treatments, the downregulation of miR-124 would allow the de-repression of synaptopodin translation and the plasticity to occur. So we actually verified that that miR-124 was downregulated by doing QRT-PCR experiments that I'm not showing here. And then we wanted to test this hypothesis by using 3 different approaches. One that I'm summarizing here. One was to overexpress miR-124 to compensate for this downregulation, and we found that this inhibited the homeostatic plasticity. Another one was to delete the seed region in the synaptopodin 3' UTR in recombinant construct that we overexpress or express in neurons. And we found that this occluded the plasticity. So those constructs, they escaped the regulation by miR-124. So they are in basal conditions, they are already highly expressed. And the final approach was to introduce locked nucleic acid sequences in neurons to selectively impair the endogenous interactions. I think this is the most direct approach. So those locked nucleic acid will bind the 3' UTR of synaptopodin to, and this way prevent the interaction with miR-124. And we found again that this occluded the homeostatic response. I'm just showing briefly the results for this last approach using immunostaining. So you had here the control experiments where neurons respond to the TTX treatment by increasing the size of the synapses, the synaptopodin expression, and AMPAR receptor accumulation. And by introducing these locked nucleic acid sequences, you can see that we have a complete occlusion and a full occlusion of the plasticity, suggesting that the expression of synaptopodin regulated by miR-124 is responsible for the homeostatic plasticity. Then we wanted to move to a more integrated system with a better organization of synaptic circuits, but in which you can still manipulate easily neurons and molecules in neurons. So we moved to organotypic slices, and we targeted CS3 pyramidal cells with a single-cell wool-cell patch clump approach. And so in this case, we used a replacement strategy. So we expressed shRNA to knock down endogenous synaptopodin. So we have here a BFP reporter. And simultaneously, we expressed a construct, synaptopodin construct, fused to RFP, so here in green, with a 3' UTR to replace the endogenous synaptopodin so that we can visualize the synaptopodin construct. So in the control situation, in the wild-type situation, you can see that again, you have synaptopodin puncta at large synapses in about 10% of the spines. And when you mutate the 3' UTR of this synaptopodin construct to prevent the targeting by miR-124, you can see that you have a large increase in the percentage of spines containing synaptopodin. This is very striking. And this is accompanied by a non-multiplicative increase of spine size, which recapitulates what we have observed before in culture neurons upon TTX treatment. So again, this suggests that the regulation of synaptopodin expression by miR124 regulates spine size in a non-multiplicative manner. And we also perform some experiments in organotypic slices to track the same cells, the same spines before and after the TTX treatment. This is what I mentioned before. And again, you can see here that, so using the same replacement strategy, and again, you can see here that the number of spines expressing synaptopodin is largely increased after the TTX treatment. So you can clearly see the same spines. And this is what I was explaining before. So here you have spines that become synaptopodin positive. So you see an enlargement of the spine size. Some protrusions remain synaptopodin negative, and they are usually the smallest ones. And you have also very large spines that already contain synaptopodin, but further increase and express even more synaptopodin. So you have this non-uniform regulation that is here very fine on a pairwise type of analysis. The last question, and this will be the last slides, was to see if this process was synapse autonomous. So can we trigger the homeostatic response simply by altering locally synaptic activity rather than spiking rates at the level of the cell body? So to do that, we used an approach that I developed previously with in a study with in collaboration with Yuki Goda. So the idea was to target CS3 pyramidal cells in organotypic slices, because we know they do recurrent connections, with patch clamp recordings. So this allows to assess the functional connectivity between the two cells, and at the same time, through the patch pipettes, you can infuse cocktails of plasmids in each of the two cells and get them expressed to visualize cells and also to manipulate the expression of any gene of interest. So after the recording, you can reseal the membrane by doing an outside-out configuration, and you can return the slices in the incubator. So the cells survive, and they express the constructs, your constructs. And then you can analyze the morphological contacts, and you can even repatch the cells to do some more electrophysiological recordings. And you can do that several rounds, so with a few days interval in between each time points. So we use this strategy to express tetanus toxin in the presynaptic terminal. And in the postsynaptic cell, we used our replacement strategy. So we found that after 48 hours of expression, by repatching the cells, we found that the connection became silent. And then we perform morphological analysis, and what we found is that spines which were in a position with a silent presynaptic terminals expressing the tetanus toxin were larger in size compared to nearby protrusions which are presumably active. And they more likely express the synaptopodin construct, which is shown here. So this suggests a local regulation of homeostatic plasticity. And then by mutating the 3' UTR to block the regulation by miR124, we found that this response was at least partly occluded by this manipulation, suggesting that, again, the regulation of synaptopodin expression by this microRNA regulate this local homeostatic response. And to finish, I would like to put this in in light of the results of another study that came out during the revisions of our paper. So this is a study by the group of Anton Maximov, in which they used a genetic model to express tetanus toxin in telencephalic neurons using a Krylog's strategy and the EMX1 promoter. So of course, there's no synaptic transmission in the hippocampus in these mice. And then they analyze the morphological characteristic of the synapses using electron microscopy. And again, so they found that a full range of morphologies for dendritic spines, and that were in the same similar numbers as compared to the wild type situations. Okay, so there is a high diversity of synapses. But then when comparing the synapses in between each category, they found that there was a large increase for the size of mushroom spines selectively in the tetanus toxin condition. The other spine types remain of similar size, resulting again in a non-uniform regulation of spine size. They also found that the percentage of spines containing a spine apparatus was largely increased in the tetanus toxin conditions. So these results are interesting because first, this is an in vivo approach, and so this supports also on results. And also in this case, so they blocked the blockade of synaptic transmission occurs very early during development. So, the synapses can develop in complete absence of glutamatergic activity. But still, we obtain the similar results. So I think that's why I think we should also think those results in terms of homeostatic plasticity. That was not really discussed in the paper. So my point here is that when you remove glutamate activity, it's not just you remove something, it's also you are doing something for the neurons as if they were stimulated somehow, which is really intriguing and when you think about it. So the conclusions are this one, yeah, and then this is the last one. So homeostatic plasticity can be implemented in a synapse autonomous manner. It depends on the initial biochemical and functional states of individual synapses, which results in non-uniform plasticity. And this process is regulated by miR-124, which regulates the local translation of synaptopodin. And the take-home message is that we have here two types of plasticity, LTP and homeostatic plasticity, that share some common rules, which are synapse specificity and also probably metaplasticity. They both rely on tagging mechanism and protein synthesis and potentially involving synaptopodin for each of them. So this is something I would like to discuss with you, how two very different induction mechanisms, so either a chronic activity blockade or a very acute hyperactivity could lead to the same type of regulation at the level of individual synapses. It's really an open question. I don't have necessarily an easy answer. And then I would like to finish by acknowledging all the people who contributed to that work, especially Sandra Dubès, who did a PhD with me several years ago. All the people from our team in Bordeaux, our collaborators, the funding agencies, and you for your attention.
So, well, you showed that there is like a molecular similarity for the LTP and the homeostatic plasticity. But do you know, like molecularly, like what is the thing that makes them different?
That's a complex question. Of course, there must be some differences, and I think one question that can be asked is what's the functional significance of what we found? So why strong synapses, and what's the significance for a spine or a synapse to express synaptopodin? So is synaptopodin a marker of some activation history? So that would be my guess. So maybe you have some occlusion between LTP and homeostatic plasticity, and maybe LTP will set the molecular tag somehow at synapses, and because maybe they store some specific information. And then when you chronically blockade activity, which is not really physiological type of manipulation, so I think it's extreme manipulation, and maybe it mimics some pathological situation. Maybe in such extreme situation where you have a chronic blockade of activity, maybe the spines which are secured and saved or even strengthened are the ones which have undergone LTP earlier. So I think one way to look at this would be to look at the interplay between LTP and homeostatic plasticity, maybe in sequence, using the organotypic slice system to see to what extent they occlude each other or not. So for now, we are missing data to answer your question, because obviously the rules are not completely the same. But maybe for the long term, I mean the late LTP, I believe that synaptopodin and probably miR-124 play a similar role as for homeostatic plasticity. But your question is quite tough, I think.
Have you overexpressed miR-124?
We have, But in the, yeah, we have, so, but in, yeah, exactly, so we have, what we know is that it blocks the homeostatic plasticity response, probably because it compensates for the downregulation of miR-124 that we found. In basal condition, it doesn't affect much synaptopodin expression. I don't know if I don't, I think I have the, yes, so it's here. So when you overexpress miR-124, you block the homeostatic response. If you look, for instance, at the increase in the percentage of synaptopodin positive spines. But if you look at basal situation, you don't affect much the expression of synaptopodin, meaning that probably synaptopodin is already highly repressed by miR-124 in basal conditions. So even if you overexpress it, you cannot downregulate more synaptopodin expression. For AMPA receptors, we found an increase in basal condition, and this has been shown also in other studies. It's probably the way to interpret that is that when you overexpress miR-124, you also downregulate GluA2, because GluA2 is a target of miR-124. And by downregulated GluA2, you promote the biogenesis of GluA1 containing AMPA receptors, which transmit larger currents. So this is how to explain this increase here. In terms of electrophysiological recordings, we didn't see much difference. But again, you block the plasticity.
So it argues that miR-124 really is tonically suppressing this. So my second question actually was really a request to maybe tell us a bit more about the spine apparatus, because my very vague memories of this are that it's not just a cytoskeletal element, but it also had membrane associated with it.
Yes,
and
sure.
There were people that were saying, well, maybe this is an ER outpost in the spine neck, which would be a place where things like AMPA receptors could be synthesized.
Yeah, exactly.
And so that things that had a spine apparatus would have a different competence to translate particular classes of mRNAs. Is that actually correct?
Yeah, this is completely correct. Actually, the spine apparatus and synaptopodin could act in many ways. So not only through the regulating the actin cytoskeleton, but also through regulating calcium signaling from the intracellular stores. So you're right, it's beyond the
So it's local calcium too.
Yeah, local calcium, and I think this has been shown by Andreas Vlachos, for instance. So this is clearly a possibility. There is also some evidence that the spine apparatus could regulate the diffusion of membrane proteins, especially it has been shown for metabotropic glutamate receptors by negatively regulating the trafficking through the intracellular domains of some receptors. So yes.
One last really quick question. Computationally, the power of homeostatic plasticity is really that it's cell-wide, so that it allows mechanisms for synapses to compete. If correct me if I'm wrong, but I think you were suggesting that the homeostatic activity detection might be synapse-specific. So computationally, that raises a number of questions. Could you elaborate on that, please?
Yes. So so spiking rate versus synaptic activity. So I didn't mention it, but there is also some other evidence showing that for glutamatergic synapses, the spiking rate might not be the parameter that is detected and compensated directly. So one way to show that, I think it's Peter Wiener who showed that, is to elicit plasticity with TTX treatment, for instance. So you would expect a decrease in the firing rate, okay? But if using an optogenetic loop, you can maintain, you can make the neuron still spiking at a similar rate. But still you are blocking activity presynaptically with the presynaptic neurons. And if you do that, you still see the homeostatic response. So even if you artificially maintain a normal firing rate, the synapses will still scale. So this is another evidence. But I think this is not true for inhibitory synapses, for instance. And again, there is a vast physiological situation for homeostatic plasticity to be expressed. I mean, it's a big family of plasticity. There's not just one way to express homeostatic plasticity. But this is puzzling, and it raises the question of how LTP and homeostatic plasticity can coexist at the same synapse. One possibility that, and I think it's an important point, is that actually homeostatic plasticity and LTP represent the same phenomenon. So maybe when you express LTP, maybe you're actually changing the threshold or affecting the set points for homeostatic plasticity. So if you raise these set points, then you can get LTP. So maybe maybe this is where the two converge. I don't know if you...
Yeah, I mean, the question is whether it's synapse-specific or cell-wide, but it has interesting computational implications. But thank you. Maybe we can come back to that later.
Yeah.
Great. Thanks very much.